Thank you so much, Shally, for a kind introduction. And it is my privilege to talk to this group of people. And it's very touching. I thought that it is not that common disease myself. But as the time goes, and my friend who is Dr. Jog, and a neuromotor specialist at London, and when I wanted to take some time from him, I found out he's so busy. One neuromuscular specialist, Southwestern Ontario, has 3,000 patients with him. This is how common it is. Also, Dr. Joke tells me, previously thought that this disease is kind of age-related, and later in the age, it happens after 70, 80, 90. And again, a terrifying fact that more and more early onset diagnosis is also showing up. So for those who never thought that it is such a desperate problem, we think it is very desperate. We have to do all we can do in our power to deal with it and make sure that our next generation and other are not as bad as that, and we can find something to help and cure. Yes, as mentioned before, 200 years, and we are still there. As I said, we have been able to send our probe to Mars and so many other advancements. Almost the latest was that they can transplant the whole head, I believe. All that advancement is done, but still, for a disease like this and affecting so many people, we are still at the same place where we were long ago. So we have to move forward. And what I'm trying to do today, I will present our research work, and I want to clarify. Next slide, please. Oh, it is here. Oh, I can go with this, right? Thank you. So I am a researcher, professor of biochemistry, PhD doctor, not MD. So I have to qualify myself here that I am not authorized to make any suggestion, any recommendation. Only health professional can do that. I will present the result of our research work we do. To us, it is very convincing, but maximum of my research reach is up to the mice and rat. Above that, all is in the hands of doctor. And we are hoping that what we are working on will be translated to clinics sometime sooner than later. With that declaration, let us move forward. I have prepared this presentation, and it's a very simple one, so please forgive me if it is too trivial for you, and also forgive me if there are too many jargons as well. I will try to stay focused on the subject. So the cells we are all assembly of cells, trillions and trillions of cells. And cells have only two jobs, that is what people thought. They keep dividing and differentiating. The same cells, we all start with one cell, long back after fertilization, and 1 to 2, 2 to 4. Some cells make our brain, some cells make our eyes, some cells make our skin, and so on. That is called differentiation. So cells divide and differentiate. What was not known before, cells also commit suicide. They die. And that is where is the laser here?
The lasers, the small green one?
Yeah, got it. Oh, this is very sophisticated. So cells have inbuilt program to kill themselves. And actually, people thought, why should there be any program to killing themselves? But it is for a very good reason. Out of trillion cells, millions and sometimes billion cells have the program, they have done their job, and now if they don't die, they may become cancerous. So as I am talking to you, many of our blood cells, skin cells, they keep committing suicide so that I am healthy. The live example of cell suicide is the people who go for sun tanning and come back, and you see one whole layer, that has committed suicide. Even if one cell from that did not commit suicide and had got radiation-induced damage, that can become cancerous. That's why. However, like anything else, if it happens too much, that also is a problem. So this is the difference between the programmed cell death or apoptosis, what the term was introduced, and necrotic cell death. This is pathological and traumatic injury when you got hit, burnt, a toxin, the cells die. And this is the programmed one. The cells do not leak, they shrink, and they pinch off as apoptotic bodies, and they are eaten up by the neighboring cells. That's why there are lots of cells dying in my blood cells, skins, and I don't feel it at all because they are taken care of it immediately. Whereas if the cells die by leakage, we get inflammation and we feel it. So that is the phenomena. What happens? If there is excessive cell death, where it should not happen, we are all born with similar number of neurons at the birth. And some neurons keep dying on their own, and we have to live with whatever neurons or brain cells we are born with. And those cells has to live as long as we live 80 year, 90 year, maybe 100 year. So those cells are very critical. We have to take care of our brain cells. And if they start dying prematurely, then we end up with the disease. So one of my research focus is, we have to go back. Research focus is to see how cells start dying in our brain, and if we can interfere with that, prevent them from dying, this excessive cell death. On the other side, as in the introduction you were mentioned, the cancer cells are crazy. They have forgotten to die. And we very desperately need to remind them that your turn is over, go, die. So our project is on both sides. Now coming to our neuronal health, a very important problem as the population ages, and also the environment, the water, food, somehow something has, and what I call the stress. I believe my father was telling, he is no more now, that when he was young, people were kind of relaxed. They were not under too much of stress. They were working very hard and making their end to meet without any problem. But they were not as stressed and panicked as young people today. And that is very important because whether your food is bad, or air is bad, that is the one thing, but if your environment under peer pressure or whatever, you are stressed constantly, that also is a bad pollution, I say. So we have to stay away from it. So all these things affect our brain, and our brain cells, as I said, have to live for a very long time. So we have to do our best to take care of them. Don't panic, don't put unnecessary pressure. Some of the evidence is that when you are very upset, your face goes red, your hands start shaking. Remember what is happening. Your brain is stressed, it is releasing all sorts of neurotransmitters, which is affecting the body. Having said that, so one of the reasons why these diseases like AD and PD are increasing is on our social system as well and under stress and anxiety. Now we do find that oxidative vulnerability, the free radical induced damage and cause cell death, that is definitely one of the reasons for this. And mitochondria is a key in this whole thing. Our brain depends for energy on mitochondria. And if anything goes wrong with mitochondria, our brain cells are in trouble. So our focus of research today is on mitochondria. In my lab particularly, we work on mitochondria. So we can do anything we can do to make it healthy and not leaky at all in the brain cells. In that way, keep the neuronal cells, brain cells healthy. And I will be talking about coenzyme Q10, water-soluble formulation. We have some other molecules which can block this bad protein, but I will not be talking about it today. Also, in cancer cells, we are trying to focus on this. Cancer cells don't use mitochondria, and yet, if mitochondria leaks, it can kill cancer cells. So, in my lab, mitochondria is called Chernobyl of the cells. It is a powerhouse. It produces energy if it is intact, and if it leaks, you are doomed. Basically, it induces cell death. So in cancer, we are trying to make it leaky. In brain cells, we are trying to protect it. So our work is focused on mitochondria. And these are the young people who are involved. In fact, there are new young people who are sitting on the back of this room. Their picture is not there. But whatever I'm going to present, the previous work is done by this group of people. We call it mice group, rat group. And we use both model systems. Coenzyme Q10 is one of the components naturally made in our body, And it is already available in the stores, but it is oil-soluble formulation. Generally, it is one of the main component of the mitochondria. And if it is little bit more in the mitochondria will make 25% more ATP. So it is all good. And we thought, why not use CoQ10 to protect neuronal cells under oxidative stress? That is our first aim. The trouble was this oil-soluble CoQ10, whatever you buy from the store, you put in the culture media, we grow the cells in the water-based culture media, it will just float on the top. It won't get into the cells at all. That was the problem we faced when I was at National Research Council in Ottawa. So we thought of solubilizing that into water. So this technology was developed at NRC, a Crown corporation, patented and transferred to a private corporation quite a while ago. And in this technology, we combined CoQ10 with vitamin E, and that was combined with polyethylene glycol. So it will make a very nice micellar formulation. This yellow thing is CoQ10 inside. Outside is polyethylene glycol, which is water soluble. This nanomicellar formulation was water soluble, and it could get into the cells. And the best part, I'm going to show you some proof, it gets to the brain as well. So we thought that now formulation is good, let us go ahead and start testing. Now, as a researcher, we want to have a model. We can have some brain cells to work on. Definitely we would like to have brain cells from human, but that is more relevant, but nobody will be willing to donate them. And ethically also it is very challenging because if you want to get some brain cells from a person who is going to donate and has written in his will, it has to be obtained immediately after death, and that is very difficult. So our search was made easier by some of the people who took neuroblastoma and teratocarcinoma. These are brain cancer cells basically, and they have established cell line. We can force those cancer cells to differentiate and behave like neurons. So that is our model to start with. So these are the brain cancer cells, and we treat them with neurotropic factors, and we just request them, please, can you become neurons? Well, chemically, we give them all signals and all the media, and under certain circumstances, they do differentiate and behave like neurons. So this was one system. And this is another system. This is neuroblastoma cells. They can be differentiated into neurons, and they do talk to each other through their neurites or axons like our neurons behave, and that gives us a tool. So now we can challenge these cells with toxins or oxidative stress and see if we can protect them. So is video working?
I know it didn't play.
Didn't work. It's okay. So this was the differentiated cell. Basically, this is neuronal cells differentiated. We threw some H2O2, hydrogen peroxide. It is a free radical, or it's an oxidative stress compound. And we throw it right on them, and they start dying. Their axons were breaking away, and they were looking miserable, basically. Now, what we did next, that water-soluble coenzyme Q10 we prepared, we incubated these cells with that and then challenged them with H2O2. And this is what happened. Again, if the movie was playing, you will see that nothing will happen to them. Their neurites were strong, axons were strong, the cell body was fine, nothing popping up and nothing happening. My boss, Mariana Sikorska, at that time told me that basically they are saying, nothing will happen to me, kind of laughing at us. This was near complete protection by this water-soluble coenzyme Q10. Very exciting. In fact, this was the movie. You can see the standby pictures. Very clearly, these two are smaller magnification, higher magnification, treated with coenzyme with hydrogen peroxide without coenzyme Q10, without water-soluble coenzyme Q10. And these are the treatment done in the presence of coenzyme Q10. You can easily see the health of those neurons. So this was very exciting to us, actually. It was 2005 we published that in NDB. Similarly, at further higher magnification, this is glutamate excitotoxicity, MSG, what we call. Again, it is not that bad, but in isolated cells, if you put excessive glutamate, that is called excitotoxicity, the cells start dying. So not only hydrogen peroxide as a stress, we use another stress, glutamate. And in those circumstances also, these cells are dying, and they were protected, very well protected by coenzyme Q10. Then we use another cell model. This is the SciY cells. These are healthy cells. When they were treated with S2O2 paraquat, I will be introducing you to in a moment. It is a toxin. There was a time people were using a huge amount as a herbicide to get rid of the weeds. And now it is banned, but we have been exposed quite a bit. That material, if you put on the neuronal cells, they start dying. You see, they are looking pretty terrible. All these bright spots, dead cells. If you do the treatment with CoQ10, they are all protected. So we have to do it multiple times under multiple conditions and with different cell types to convince our reviewers and publishers that, yes, it is true. Because sometimes something may work for only one cell type, doesn't work for others. So these are the standard scientific practices. Now, we did another thing. This is the mitochondrial staining. We stain the mitochondria with a stain called JC1, and we found out that if we treat these cells with paraquat, all those staining disappear, which means mitochondria are being targeted by the toxin. So if the mitochondria are dysfunctional, the cells are dead. This coenzyme Q10 were doing a remarkable job in keeping the mitochondria intact. That was another very exciting find. It will become more clear in this one. So all the red spot you see here, it represents mitochondria. And if you treat them with hydrogen peroxide, the cells collapse and those nice red spots disappear. They all collapse. But if you do the same treatment in presence of CoQ10, the mitochondria remain very healthy. So this was our starting point, that this material in the Petri dishes on neuronal cells was able to offer near complete protection against different toxins. So where to go next? That was the question we asked. Now the question was that, can we use this material in an animal model and protect the brain cell, because that is the next step, more physiological. Many things work in Petri dishes, but they may not show the same effect when you go to the animal model. So that was our next job, to test it. Now I'm coming to the Parkinson's disease. Again, it is nothing new to this crowd. It was very nicely introduced in a video there. Whoever made that did a very good job. And as the literature stands today, we all have, if we are all healthy and perfect, at age of 15 years, let us say, when we are very young, we have 400,000 substantia nigra neurons. They are called dopaminergic neurons in our brain. It's a small area, and that is where these cells are there. This is the healthy brain, basically. 400,000 when we are young, very young, teenagers. As we grow, we lose them slowly. So my students have definitely way more neurons than me, particularly those, and as the time goes. But there is not much of effect of those slowly dying cells as a part of aging phenomena. In a 95-year-old person, there might be little bit age-related tremors, and if you take the count of substantia nigra neurons in that gentleman or woman, it will be approximately less than 50%. So would have lost. Unfortunately, in the case of Parkinson's patient, that degeneration is way more accelerated. The cells start dying in this region very fast, and at the time of diagnosis, when you really see the symptoms, half of the neurons are there. You have only 200,000 neurons. So this disease is definitely correlated to the degeneration or disappearance of substantia nigra neurons, for sure. These neuronal cells produce dopamine, but that is not a big deal. We can give you the dopamine. Dopamine can do the job. Actually, that is the only symptomatic cure we have at present. But we need them to survive. More than making the dopamine, those cells also communicate with other neurons to coordinate our functioning. So if those neurons disappear completely, we keep giving you dopamine that we can take care of it, but still it doesn't work. Again, neurologists tell me, the doctors, that if you can save only 20% of these neurons, we can make the person almost lead a very simple, normal life. Because we need very minimum amount of those neurons. We can give dopamine, and those little bit neurons are there. They can coordinate the function, and life can be normal. And that is what the thirst today is for research. We know that when the person is diagnosed, whether it's Alzheimer's or Parkinson's, it is a progressive disease. As the time goes, it becomes worse and worse and worse. It is a very dire situation. You are seeing a child walking towards a well. He's going to fall. We are all watching it go towards that and unable to do anything. In today's doctor's purse or doctor's bag, there is nothing which can halt the progression of the disease or protect these neurons. And I hope people are working very hard. They could find something where it is not symptomatic relief, but at a causative feature where we can block the progression by blocking the death of the neurons. And of course, the symptoms of all these things, as described in the video, I'm not going into detail, everybody knows. So what can we do? So this is some kind of environment and genetics, how Parkinson's is caused, and I just said that they don't know how. This is true. There is some genetic element coming out to be, but it is very, 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 very little. By and far, more than 90% of the incidence of Parkinsonism is environmental related. There was some papers, actually, there were some studies where the paraquat, I told you, the herbicide exposure, and the incidence of Parkinsonism, there was a correlation in some population where too much of this was used. But still Parkinsonism occurs in the population who have not been exposed to paraquat. It is just that it will be 6 to 8-fold higher in the people who are exposed to. So still we don't know, but definitely environment plays a role. When I say environment, of course, it is any toxin or bad pollutant which can do this, as well as environment is our family, our society, and I told you before, the anxiety and depression or the pressure of peer pressure. Those, to some extent, are responsible. Also, a physical damage to the brain. Some people deliberately would like to hit their brain. The boxers, I believe, they do that. And Muhammad Ali is one of the classic example of the so many bouts he had on his head. He developed Parkinsonism. So those are the reasons. And these are some of the genes involved in this the disease. That's what the current research is. Now, how many people know about Toby in this crowd? Not many. Toby was a 19-year-old young man who was diagnosed with Parkinsonism. The single person, actually a drug addict, who led to 3 discoveries in Parkinson's area. We did not have any working model with animal in those days. Before 85, if you want to study Parkinsonism in the rat or mice model, there is nothing available. How to create that those neurodegeneration in the brain of animal so that we can study, that model was not there because people did not know at all how Parkinsonism can be created in the animal. Well, we don't want to create that, but to know how it is happening in human even. Now, Toby was 19-year-old. He was in the jail for drug-related problems. And when his mother went to jail when he was released to accept him, receive him, he was having the tremors and the typical symptoms of Parkinsonism. But she didn't know anything about it, poor lady. She took, thanks to California, their health system is still kind of semi-private. There is a government healthcare system in California still. So she could manage to go to the clinic, and there his doctor, who was a neurologist, she was almost astonished that how come she never saw a Parkinson's symptoms before 50, before even early onset is 40. Michael J. Fox was diagnosed at the age of 39. But 19-year-old, no, she never had any case like that. She was intrigued. So she wanted to inquire what did he do. Of course, he was taking heroin. And not the other heroin. He and his uncle were making a synthetic heroin, some crazy things they do at home. without realizing that that has got a contamination of a compound. That is called MPTP, that is the name of the methylpyridinium. That was the compound which was in heroin. Maybe that contaminant was causing Parkinsonism. She kind of wrote an article in big journal Science. People use that compound can be synthesized, the MPTP contaminant, injected into mice, and lo and behold, mice behave the same as Parkinsonism. So this addict, heroin addict I call, was the person to give us a model to work on animals, that how it can be created in animals. MPTP is the causal features. Number one discovery. Number two, he was given levodopa in those days. I'm talking about early 80s, 85. At that time, the dose was not very standardized. So they gave him some dose, and it was kind of higher dose. At higher dose, levodopa can cause hallucination. They had no idea that it causes that, till they gave him a pretty good dose. And after that, he went and robbed the bank or went and broke some convenience store. He was arrested again. And in the court, when he was asked, why did you do that? He said, somebody was telling me in my ear to go and do it, which is the classical definition of hallucination. So they realized for the first time that no, dose has to be tightly controlled. That was the second discovery. Then they started working on the doses, the side effect of levodopa. And the third one, he was the first person to undergo deep brain stimulation. The new surgery these days, which is very effective in many people, basically. So he is almost normal, and he went through the deep brain stimulation. So three things were discovered with one subject or one patient. So as I was telling you, this is the MPP, that's contaminant in heroin. And thank God for that, there are not that many people taking heroin. And those who are taking, they are taking a good stuff, I believe, not with contaminated stuff, I hope. But without realizing, we found out the paraquat. This is the herbicide used in the fields. It has very similar structure as MPP. And that is what we focused our study on. So this paraquat, when we used in the rat, that caused the Parkinsonism too. And not surprising, because it is very similar. Only trouble is, this is not available to everybody, thank God for that, but this one was almost forced on us by agriculture, by using herbicides without knowing it. And in early 2000, it was banned throughout. Still, there are some countries who are using it. Okay? And there are some compounds, even there was a particular cup syrup taken off the shelf in 2003. That cup syrup also has a compound, something similar to that, and they found out and removed it from the shelves. So we use these two as a model in the animal system. So this is our research objectives, and we went ahead and do some studies, and then I'm going to share some results. So before we go ahead and try this water-soluble coenzyme Q10 on the animals, the one question will be always asked: whatever we are giving to the rat or mice, is it absorbed in the body? And if it is absorbed in the body, does it go to the brain? These are the normal pharmacokinetic experiment we do for bioavailability. So that was the first thing we did. So we gave the mice or rat, both cases, orally some of this material, coenzyme Q10, water soluble, and we found out within 1 hour it is absorbed from the intestine, goes in the bloodstreams. And as the time goes by, it starts going down in the blood, and it starts getting into the tissues. So from the blood, it goes to the brain, liver, heart, everywhere. So the amount in the blood goes down, and amount in the tissues goes up. And this is what we want. If we want a material to get into the body and get into the brain, we have to show that. And this is what we show that. In the brain, it goes quickly. In fact, if you wait longer, it starts going down there also. So if you take it, it goes to the brain, but does not keep piling up. It starts getting recycled as well. It goes down. One thing. Number two, the mice were pretty fine. No problem in terms of walking and running around, no weight gain problem. These are the things, and we also did some toxicity experiment. We have to prove to Health Canada and to the health agency that this material is not toxic by itself. So it was pretty healthy. And that is not surprising. CoQ10 is one of the natural products made in the body. What we are doing we are just increasing a little bit of this amount in the brain by giving outside supplements. So that was done. Then we had this experiment. Many of the problem, people develop a model which is an acute model. You give an injection, there is a huge amount of cell death, and then try to rescue them. That is not physiological. Parkinsonism is a progressive disease. Once it is observed, slowly those neurons keep dying. So we have to develop a model of progressive disease. For that, we took these 3 groups of animals. One group was control, of course. We gave them this paraquat injected in their body. And immediately after 5 injections, we sacrificed to see how much damage is there. And then other batch, we waited for 2 weeks, and then 4 weeks, and keep looking at the brain cells, those substantia nigra neurons. And if there is a progressive decrease after the last injection, which will validate the model. And this is what exactly we did. So these are the healthy neurons, pretty good. And this is immediately after the injection, and then it is after 15 days, and this is after 4 weeks, 30 days. The number of neurons in the brain, this is pretty good. It goes down a little bit, further down. Remember, this is where the injection was stopped. So after the toxin was injected, following that time point, as the time goes by, more and more neurons were being lost. So now we have got a window here where we can interfere and block the damage right here. So this is how we did it. And at this time, we saw this picture, but all the agencies, the granting agencies, the reviewers, they said, no, you have to count those neurons. And you cannot count it manually. There was a machine in the market, it's called Stereologer, which can automatically count the neurons, which is more believable. Otherwise, if I count it, I might be biased. So it was good. We did not have any funds for it. And thanks to Mr. Joseph Sachi from Windsor, you might have heard about him. He also made a donation to carry out our work on natural product with Parkinsonism. It was so nice. We did not even approach him. He just saw a news story in the paper, and his daughter called me, said that we will help you. And Mr. Joseph, he himself is a patient of Parkinsonism. He donated in 2009 $50,000 to acquire this instrument for us, which was very lucky for us. So of course, with that machine, again I must, Dr. Cohen is here, me, and of course in 2009 I had more hairs. And these are the students working on the project. And we could count it. And indeed, whether you take percentage of neurons or count those neurons, you can see the progressive decline of those neurons. So if we interfere here, we cause the damage right after 5 injections, and we start giving the CoQ10 right here, and if our hypothesis is correct, then that compound will keep the neurons number same. It won't go down, right? We can't do anything between this and this. We had done that before. That is called prophylactic studies, where we give the CoQ10 first and then injected the toxin. Of course, it's protected it. But we are not expecting that everybody will take it, thinking that they might get Parkinsonism. We are here trying to do a therapeutic intervention. Once you have been diagnosed, once we know that 50% neurons are not there anymore, can we save the rest of the 50%? That was the whole idea. And for that, we planned the experiment, and we did that. So this is a simple experiment, where this is the control number of neurons, and this is paraquat injected for 4 weeks. So after 4 weeks, this is the number of neurons, and after the injection, if we give CoQ10 for 4 weeks at the same time, you see the number of neurons here are definitely more than this, not exactly like this, because already some neurons are lost. This CoQ10 will not revive the neuron. It is not that miracle that it will bring the dead neuron to life. No. Whatever dead is dead. But whatever are alive, they will be protected. That is the hypothesis. And indeed, we counted it, and you see here. So this is the 17%, 0.6% neurons here are dead after the last injection. And if you wait for 4 weeks, 41% neurons will be dead over here. But if you put our material CoQ10 over here, only 16% neurons are dead, which is similar to this. So whichever are alive, they're alive here as well. And of course, this one was done again with placebo as well. So if you do the only carrier solution, only vitamin A, this is the carrier solution, there is not much protection. It has to be the water-soluble coenzyme Q10. So then we went ahead and did another thing. How long we should give coenzyme Q10 to the rat to protect that neuron? So we did 1 month and 2 month study, where we induced the toxin in the animal, and after that, one group of animal was kept 4 weeks and 8 weeks, 1 month and 2 months without CoQ10. The other group, we gave CoQ10 only for 1 month, and then remove the CoQ10, let it go without CoQ10 for another 1 month, and see if we stop giving CoQ10, the number goes down or it stays the same. Or keep giving CoQ10 all the way till 2 months. And that was the experiment planned. And this is the result, where we have this one, which goes on for 2 months, and there is a drastic loss of neurons, more than 50%, over 2 months. Now we interfere and keep them for 4 weeks, and after 1 month, we remove CoQ10, no CoQ10. We did find that they were protected, but not as much as we keep giving constantly CoQ10 all the time for 2 months, which indicated that the toxin is still there in the brain, and if you remove CoQ10, it might start acting again. The neurons may start dying again. So, of course, this requires that we do for not only 2 months, 6 months, and 1 year, and we are hoping to do that in our further work. But yes, CoQ10 is non-toxic, it is a new food supplement. Why to stop it? People can keep taking it if it works. Again, I cannot say anything about how it will work in human being. But that is the projection. And of course, we counted it, and this is the counting results. So here we see 50%, 47% neuron loss in 2 months. If you keep giving CoQ10 for 1 month and remove it, then 28% are lost, definitely more than this. And if you keep giving CoQ10 all the time, then the loss is only 14%. And of course, we can't do anything with this because we started our treatment only after the last injection. We give 5 injections and then we start. So definitely, this is pretty much with the hypothesis. Now, in the brain, we can see that neurons are protected. Only 14% lost because when we interfered, 14% were lost, rest of them were good, and we keep that same amount all the way if we keep giving them water-soluble CoQ10. Very good. But does this neuroprotection translates into behavior? Sometimes we cut the brain, say everything is fine, and still there is no effect on the motility of the animal. So we have to do behavioral test. That if the animal is behaving normal as well, then it will be a good test. And that is where Dr. Cohen's lab comes in picture. It is a very hard work for them. They train these rat and mice to walk onto a platform and keep watching their slippage of the leg. The leg slips if there is a toxin-induced damage. It's kind of similar to Parkinson's symptom. So the number or percentage of the leg slippage is our outcome, that if it has got the Parkinsonism-like symptoms. So this is white is the control, no lesion, healthy animal, and it makes only 1 slippage in the whole walk. So that is the normal one. Now, if you inject it for 4 weeks, the paraquat into the brain of this, after 4 weeks, if you measure it, there are many neurons are lost. It is going to display the Parkinson's syndrome, and indeed it does so, close to 3 slippage in the same walk. Now, if we give coenzyme Q10 for 4 weeks, for 1 month, and measure it, yes, the slippage come down, which means whatever we saw biochemically in the brain is being translated into behavior. However, if you go for 8 weeks, remember we removed the CoQ10 after 4 weeks, and there were more neurons dead. Somehow the slippage remains the same. So if you remove it from the system, there is not much protection. But if you keep giving them for 8 weeks, there is definitely protection. And of course, this is unprotected one. Sorry, if you keep giving for 8 weeks, there is complete protection. They behave as good as control animals. But if you remove it for 4 weeks or 8 weeks, if you don't give CoQ10, their behavior will be almost like a diseased animal. So this one was complementary to what we were showing histologically. So when these two, behavioral and histological data, talk to each other, it becomes very important. That is why these studies were published in Neurobiology of Aging. So this one was already done. So this is our studies results from the rat model. Again, the funding agencies and the journals who publish our work, they said, well, it is working with paraquat in the rat only. Will it also work in mice where we give MPTP? They wanted us to use another model. And of course, we went ahead and did that. So this is the mice, apparently listening to some music. So we did the same thing. We stain the neurons, and we do find there is loss of neurons, and if you give CoQ10, there is pretty good protection. And I'm just repeating the same thing. In this case, in mice, we injected MPTP, the Tobis heroin addiction, that compound, toxin compound. And we give every day 6 injections in a very narrow window. It does causes acute damage. And you can see when we injected, they are dead for 2 months. And this is the higher magnification, lower magnification. And if we keep giving them coenzyme Q10, there was protection. So this one was another test, the same thing over 8 weeks, keep protecting. All the neurons are protected only when you give this supplement for all the way to 2 months, if you're not stopping halfway. If you stop halfway, they start dying again. Exactly finding the same thing as you found in the rat. So this is having 2 models, both of them showing the same results. So that is very encouraging. And this is the behavioral result, again slippage of the feet over here. This is what we monitor, and it was similar to what I told you before. This is another result we found, that astroglia, which are neuroprotecting cells in the brain, somehow they get activated in the animals who take CoQ10. And our hypothesis is that these activated astroglia, which are neuroprotective, they must be offering some protection. Then they said, okay, you have done in toxin injection, why don't you use transgenic model? These are the mice in which one gene is defective, and because of that, they develop this neurodegeneration like Parkinsonism. So there we combine neurotoxin along with this gene mutation and try to use that model. That was the third model we used. And results were again pretty clear, black and white. These are the healthy animals. These are transgenic animals which have got the bad genes, and we give a small amount of MPTP, and you see the neuronal loss pretty significant. But if we keep them on CoQ10, then get protection. Now, I should tell you, these mice were immediately put on CoQ10 when they came in our lab, because they had got the bad genes. They are going to develop the disease. So we have to interfere right away. So this treatment is called prophylactic. So even before any toxin injection or before the symptoms appear, we give them CoQ10, and that diet protected the neurons from dying automatically. And of course, we counted the neurons. If they're unprotected, they keep dying. This is the placebo, 50% neurons dead. And if you keep them on CoQ10, only 19% dead. Again, not complete protection, but pretty good. Behaviorally also, we saw the same results. If they are protected here, their slippage is far more lesser than the one which are injected with paraquat without protection. There is no protection here, and this is with CoQ10. This is W means water. This is Ubisol Q10, that is coenzyme Q10. So their behavior was also translating in the same way. Now, everybody at this stage, this project was funded by Michael J. Fox Foundation as well as CIHR, and we thought, why not go to clinical trial? And at that time, a clinical trial was already underway with oil-soluble coenzyme Q10, already in the market. It was funded by National Institute of Health, NIH, in the United States, in multiple centers. And people were given for last 2 years, and they found out there was no effect at all. And the clinical trial with oil-soluble standard CoQ10 was dropped halfway through because there was no effect in placebo and treated patients. And that was bad for their project, but it was worse for us. Now, wherever we go to take it to clinical trial, they say, oh, this is the CoQ10, it doesn't work. Then we have to get into the literature. We found out that oil-soluble coenzyme Q10 was given to mice, indeed. It showed some neuroprotection, but only at very high concentration. 400 milligram per kilo per day in the mice it was given. If I translate that to a patient like me, 70 kilogram, I had to take close to 28 grams of CoQ10 per day to see any protection. That is just not possible. Maximum amount can be given to a patient is only 1.2 gram. So when that, I spoke to clinician and I said that when you are giving 1/10 or 1/100th of the dose, then how can you expect it to work? So we compare our dose with this formulation is 6 milligram per kilogram per day, which will be 420 milligram for the whole day for a patient. It is feasible. And this is what we are trying to convince right now to the company who has got the technology transfer from NRC to go for clinical trial. There are foundations who are able to help, but clinical trial, they have to go to spend some money, and if it works, they can make big money, but somehow there is some inertia there for the companies to go for clinical trial because it is expensive study. Still, we are working, hopefully it will get into clinical trial. I'm also trying to convince the company to bring that material into the market, over the counter, without any claim. So that if I know that this is in the market, I can go and buy it from the store and take it, rather than the oil-soluble one. It is not in the market yet. They are working on approval from Health Canada to bring it out in the market. So this is the difference. There you go, there you go. 280 capsule versus 1 capsule. This is how the student made a cartoon. So we are hoping that this material can get into clinics and offer the same protection what we see in the mice. What we saw in the mice and rat and the petri dish is undoubtedly very clear results. There are sometimes you get neuroprotection, it is kind of maybe, may not be, gray area. But then here we have very clear indication. Our hope is that if this formulation gets into human body, hopefully it will start protecting whatever leftover neurons are there, and it can stop the progression of the disease. At that stage, just this year, in March, again, a Sachi family, Mr. Sachi, again, he is now in the late stage Parkinsonism, he made a commitment. He said, why don't you also combine with CoQ10 some natural extract? There is a natural extract called ashwagandha. It is an Ayurvedic medicine being used for years and years, decades and centuries for brain-related diseases. There is no indication of Parkinson's disease in Ayurveda, but people were taking it. There was some literature showing that root extract, ashwagandha root, not dandelion, please, okay, that has some good effect in these diseases in some models. So the new project, that is where our students are going to start this summer, is to have CoQ10 and the ashwagandha extract both, and if that helps, then people can, again, that is over-the-counter material, can buy, you can just grind it and make extract, hopefully. So that is the new thing we are starting, hoping that it will be of some use. And of course, I am the only communicator here. Most of the work is done by these young people. And this is my whole lab during the mango party. We have, and this is our almost yearly ritual. And many of the students have already graduated. And this work in particularly was done by Kritika. And Kritika, it must be Annie, I can see, and somewhere. Do you see Kritika somewhere on the back? One of the graduate students. Oh, yeah, here it is. Here she is. She has gone to University of Toronto to do the postdoctoral studies, and many students from Dr. Cohen's group were also working on it. And I just communicated the results. And of course, our animals, we treat them very well. This is Kritika again. And these are the undergraduate students. This is a postdoc. So these rats, actually, they almost behave like pet animals. You can't imagine that you are injecting toxin to the rat and they are not crying. They're better than babies, basically. They just pet them, hold it, and they will inject in without even squeak. So they're very good, and we have to take good care of the animal because we are doing the behavioral study. If they are anxious, they are afraid, then they will be slipping their leg all over, right? We have to bring them to calm stage, like they are very comfortable, then let them walk on the platform and watch the leg slippage. So in order to do that, we take really good care of these animals. And of course, the mango party. Back in 2009, I had to bring this picture because my parents were there at that time. My father, who is no more, his objective was that if anything can be done for age-related disorder, it would be appreciated. He had a belief that there is God in everyone. In some people, the God is very obvious to see. And Mr. Sache was one of them, according to him, who was so generous, who was not even approached. He saw the problem and he came out to. So again, I believe there are God like that in everyone, and it is because of all your help and all the community support we could manage to do all this work, so we are grateful to them. And of course, Dr. Sikorska was my previous boss in NRC. We together developed this formulation, and she was my collaborator. She's retired. And of course, Dr. Cohen is my collaborator here at the university who does the behavioral work. Without him, we couldn't do much of the behavior part. And of course, the funding. Of course, Sachi family, as I mentioned, Michael J. Fox Foundation, Canadian Institute of Health Research, and University of Windsor. We are hoping that we will bring something, and it is just we need some collaboration, cooperation, and help from industry and the clinicians. So remember, all we can do is to go up to the animals. We have very convincing results, but it is of no use if it doesn't go in clinic. We are trying very hard, and we are hoping that something will come, and we are hoping that in the next 5 to 10 years, there will be something to interfere in the progression of this disease, and people can lead to normal life. That is our hope. With that, thank you very much. This is how the platform is there, if you are wondering, to find the leg slippage. It is not a very flat, big platform. It is very tiny. But they do that, and thanks to all of you for your patience. I am ready for any question. Thank you.
So if anyone has any questions,